Hey all, so today we're going to be talking about C1.2, cellular respiration, and we want to be thinking about how is energy distributed and used inside cells. We're going to be looking at page 361 to 369, and we're trying to think about interaction and interdependence. And so how are the processes connected to one another inside a mitochondria in order to generate ATP. So firstly, thinking about ATP, we need to recognize that this is the energy needed in the cell to make things happen. And if you didn't have this energy in your cells, even just for a few minutes, then you would die. So this is for making molecules, it's for active transport, it's for movement. So it's critical to life. It is a nucleotide because it's made up of adenine, ribose, and 3 phosphate groups. And cellular respiration is where mitochondria use oxygen and glucose to make ATP. And these phosphate groups here, they have a negative charge on them. ATP being broken down into ADP produces a tiny amount of energy, but it's enough for the cell to work. And if it was making more energy, actually too much heat would be generated by the body, and then you would rapidly overheat. So the way that the energy is made is by this bond breaking. From when one of the phosphate groups gets broken off, that produces a little energy. And then ADP can be rephosphorylated back into ATP, and that's a cyclical reaction. ATP is soluble in water, but it cannot pass through the phospholipid bilayer, so that means it needs to be used inside the cell. So cellular respiration is occurring in mitochondria, and that's what's going to be generating this ATP. ATP has more potential energy than ADP, so some is released when this conversion happens, meaning that ATP is converted into ADP. But energy is needed to make ADP go back into ATP, and there's specific tools, adaptations inside the mitochondria that can allow for a concentration gradient buildup, and then that flow of ions back down the concentration gradient generates the energy to rephosphorylate ATP. And in order to generate that concentration gradient, mitochondria need hydrogen ions. And so the way that they get those is through oxidation of proteins, carbohydrates, or lipids. Oxidation means like pulling off that hydrogen ion. In the case of photosynthesis, they use water for that hydrogen ion. And in the case of chemosynthesis, which is right here, they're using sulfur compounds or chemical compounds for that hydrogen ion. So the overall process of cellular respiration uses glucose and oxygen to make ATP. Oxygen and carbon dioxide, so oxygen is going to enter and CO2 is going to move through diffusion into the cell, and these processes are interconnected. So CO2 is a waste product. It's going to move from the mitochondria into the cell and then back into the bloodstream, and oxygen goes the opposite direction. So for SL level, you do need to know the difference between anaerobic and aerobic respiration. In order to make lots of ATP, oxygen is required. That's because you don't really need to know this for SL, but HL students, you'll need to know this in way more detail. But oxygen is the final ion acceptor. And so without oxygen, then the flow from high concentration to low concentration of hydrogen ions can't happen. It gets clogged up. So anaerobic respiration is happening without oxygen. That happens in the cytoplasm of the cell, not in the mitochondria. And this process would produce, anaerobic respiration would produce about 4 ATPs, whereas aerobic, going through the whole process, produces about 32. So obviously aerobic respiration is more efficient and it's going to make more energy. Yeast anaerobically respires differently than humans do. And we can both anaerobically respire, but yeast, you'll know, probably is used for making bread. It's also used for making alcohol. And it's a byproduct of this anaerobic respiration process for yeast. And so the process for yeast, when they anaerobically respire, they're going to produce ethanol, which is alcohol, and carbon dioxide. And in the case of humans or mammals, they're going to produce lactic acid, which is also sometimes called lactate. And lactic acid will slowly reduce muscle performance. It needs to be broken down, and that's going to require oxygen to break that lactic acid down. So that's called an oxygen debt. That's about the some extent of what you need to know for that. But that oxygen debt is how much oxygen is needed in order to break down lactic acid buildup. With yeast anaerobically respiring, if you're making bread, the carbon dioxide helps to fluff the bread product so that it's fluffy when it's baked, and then the alcohol will just evaporate when the baking process is occurring. If you were making beer, for example, yeast would be used to make the beer fizzy. That's where the carbon dioxide is coming in, and then also generate alcohol. And then, so this is the last slide for this unit or section of this unit. Respirometers are a way of testing how much respiration is occurring. And so you can put bugs. You would have to make sure, if you were testing this ethically for IB, you would be taking care of those bugs and not killing them. But you could put bugs, or you could put seeds, which would be more ethical in here. Seeds also respire because prior to them germinating, they can't conduct photosynthesis because they don't have leaves. So they're going to respire, and the carbon dioxide that they produce in that respiration process is going to be absorbed by potassium hydroxide solution down here. And so if you imagine these bugs or seeds are taking in oxygen, and the replacement carbon dioxide that would come out is instead going into the solution, that means the volume of gases in this tube is going to decrease. And if the volume of gases are decreasing, then the pressure here is going to also decrease because there's less molecules in there. With a pressure decrease, the liquid inside the tube, this tube here measuring device is called a manometer. So that's going to rise towards the low pressure area. And you would then be able to measure through these graduations on the manometer how much change is occurring over time with different seeds, for example, different temperatures, different environmental conditions. All right, so to summarize here, mitochondria make ATP using oxygen and glucose. Carbon dioxide is a waste product of aerobic cellular respiration. Anaerobic respiration occurs when oxygen is not available, and it's going to produce lactate in mammals or ethanol and carbon dioxide in yeast. Anaerobic respiration makes less ATP, and it's only happening in the cytoplasm, not in the mitochondria. And then lastly, respirometers are lab equipment to measure cellular respiration. All right, so that's it for this video. Thank you for watching.